All right, welcome everyone to the HBL seminar. So we'll get started. So today we're joined by Dr. Ellen Koch, who's a postdoctoral fellow here in the HBL. So Ellen started her academic career at UBC, where she completed her Bachelor's of Science with honors in biology and psychology. And her honors thesis research focused on mapping neuronal connections in fruit flies. She then went on to complete a PhD at UBC in neuroscience, where her dissertation research examined brain signaling and motor kinematics in a mouse model of Huntington's disease. And she's currently a postdoc with doctors Tyler Clough and Sean Duclos at Foothills, and her work uses robotic technology to examine sensory motor adaptation following stroke. So a fun fact about Ellen is that she has been a yoga instructor on the side for over 10 years. So today Ellen is going to be sharing some of her work from her PhD on neuroimaging and motor kinematics in a mouse model of Huntington's disease. Take it away.
Awesome. Thank you for that introduction. So the topic today might be a bit different than some of the topics normally covered in this seminar, but I'm talking about my PhD work where I looked at motor learning, kinematics, and in vivo neuroimaging in a Huntington's disease mouse model. And just a little bit of background on Huntington's disease. So this is a neurodegenerative disease and a movement disorder. And in addition to severe motor deficits and motor symptoms that people with Huntington's disease experience, they can also experience cognitive and psychiatric symptoms. And there is currently no cure for Huntington's disease. And the motor symptoms include involuntary dance-like movements, termed chorea symptoms, motor coordination deficits, and impaired motor learning. And I'll talk about all of these in a bit more detail in a few slides later on in the presentation. And Huntington's disease is a genetic disorder. So it's caused by a single mutation in the Huntington gene. And this is an autosomal dominant mutation. So that means that when your parent has the mutation, all of their offspring have a 50% chance of inheriting the disease. In particular, the mutation that happens is called a CAG repeat expansion. And without going into a ton of detail, what this means is that in the normal Huntington gene, there are these base pair repeats of CAG repeats in the DNA. And there's a certain number, usually less than 26 of these in the normal gene. When people have Huntington's disease, there are more than 40 of these repeats, leading them to develop the disease. The average age of onset of Huntington's disease is 40 years old, which is relatively young for a neurodegenerative disorder. And the disease is diagnosed when people start to show signs of chorea, which is that involuntary dance-like movements. So that's when usually people are diagnosed. And I'll just talk a little bit more about the genetics here. So normally people have, as I mentioned, less than 26 repeats, or they might have 27 to 35 CAG repeats in their DNA, and they won't be affected by Huntington's disease. But if people have between 36 and 39 of these repeats, then they might have either a milder form of Huntington's disease or not have the disease at all. But once people have more than 40 of these repeats, people will have the disorder. This is important because the number of repeats that they have is correlated with their age of onset. The more repeats, the earlier they'll get the disease. So the 40 is the average, but it can be as early as, in very severe cases, children can get Huntington's disease when there's a very high number of repeats. The good thing, kind of good as far as research goes about Huntington's disease, is that we can identify people who are going to develop this disorder by genetically testing them for the disease. So because of that, we're able to research people who haven't actually developed chorea yet and look at earlier signs of Huntington's disease. And studies looking at that have found that up to 20 years prior to the onset of the disease, subtle impairments in motor, cognitive, and psychiatric domains have already started to appear. And then one other thing to mention here is that this is a progressive disease. So after the onset, the symptoms will worsen until death, and people usually will die from the disease 10 to 15 years after the diagnosis. Okay, so I have a couple of videos showing you what chorea looks like. So this is a Huntington's disease patient with chorea walking, and you can see a lot of issues with their gait and these involuntary movements, especially in this case in his hands, as well as a little bit in his face. You can see also these finger movements as well that are happening. And then chorea can also affect the face and the head and the neck very severely. So this is an example of a woman with Huntington's disease showing severe chorea symptoms in the face and these movements of the neck and the head and the tongue as well. So chorea is kind of the characteristic symptom that Huntington's disease is known for, but there are an array of other symptoms as well. At early stages of the disease, people will suffer from other motor symptoms, including an inability to perform fast alternating movements like finger tapping, abnormal eye movements, and gait impairments. And then at late stages of the disease actually progresses to more of a Parkinsonian phenotype. So people start to have difficulty initiating movement, slower movements, rigidity, and then very prominent gait and balance impairments. And often people end up being wheelchair bound at the end stages of the disease. One thing I was particularly interested in my PhD work was motor learning. And motor skill learning has been found to be impaired in Huntington's disease as well. And this starts during those prodromal, which is that stage before the diagnosis, up to 20 years before the diagnosis, and at very early stages of the disease. So in this study here, they looked at a motor skill learning task. This is a bit of a complicated figure, but just the basic protocol for this task was a task where the participants reached to targets that would randomly appear. There were 8 different targets that appeared. And they were instructed to learn the sequence and to try to anticipate which target would appear next in their movements. What they found in this study, and other studies have kind of replicated this with similar tasks, is that in prodromal HD and very early stages of HD, there is a significant deficit in motor skill learning. And if you look at the estimated number of years before the onset of the disease in these participants, the closer they are to diagnosis of Huntington's disease, the worse their motor learning was. So we've talked a lot about the clinical motor symptoms of Huntington's disease. What about what's happening in the brain? So HD causes neurodegeneration in very specific areas of the brain. The most degenerated area is the striatum, which is an important part of the basal ganglia circuit. You might have heard of this in a motor learning class or in a neuroscience class. This is a circuit in the brain that's important for controlling movement, motor learning, and other behaviors. So here, I'll just use my pointer on the screen here. Here is a cartoon of a normal brain, and the basal ganglia, including the striatum and other structures, is shown in gray here. And then this is an example of somebody with Huntington's disease. And you can see here that there's severe atrophy of different structures within the basal ganglia, which causes the ventricles in the brain to become enlarged. And then also the cortex all over is degenerated as well, and much smaller than the normal brain. So I'll go through the different pathways in the basal ganglia briefly here, which will help to understand some of the symptoms of Huntington's disease. So these are some of the different structures in the basal ganglia. The striatum is the signaling hub that receives a lot of inputs from other structures as well as outputs to other areas of the brain. And the striatum receives excitatory input from the cortex, as well as dopamine input from the substantia nigra pars compacta. There are 2 main pathways in the basal ganglia. And within the striatum, they are the two pathways have two different cell populations that they signal through. So one of them is the D1 neurons, and these are called D1 because they express dopamine 1 receptors. The other is the D2 neurons, and they express dopamine 2 receptors. So two different cell populations. And then the first pathway that I'll talk about is called the direct pathway. So the direct pathway signals through the D1 dopamine receptor neurons to a structure called the globus pallidus, as well as the substantia nigra reticulata, which then signals to the thalamus. And the most important thing to know is that the end result is excitatory input to the cortex, activating the cortex, which leads to voluntary movement. So this pathway is involved in initiating voluntary movements, and sometimes it's referred to as the GO pathway as a result. The other pathway is the D2 pathway, also called the indirect pathway. So this pathway goes, there's dopamine input to the D2 neurons, which then connect to the globus pallidus external, the subthalamic nucleus, back to the globus pallidus internal, substantia nigra reticulata. And then what I want you to know about this pathway is that the end result is the opposite of the GO pathway. It ends with inhibiting the cortex, which results in reducing voluntary movements. So the GO pathway or the direct pathway promotes voluntary movement. The no-GO pathway or the indirect pathway inhibits voluntary movement. And the classic model is that both of these pathways compete with each other to produce the correct movements for a situation. More recent research has shown that this isn't quite so simple as just a GO and a no-GO pathway, and there's a lot of other stuff going on, but this is still a simple way of looking at this to explain some of the symptoms of diseases like Huntington's disease. So in early Huntington's disease, it is specifically these D2 neurons that degenerate. And when this happens, this corresponds to the chorea symptoms. So without this no-GO pathway inhibiting voluntary movement, we get the excessive voluntary movement seen with chorea. In late Huntington's disease, it's both of the pathways that are now affected. So the direct pathway neurons as well. And so without both of these pathways, Huntington's disease patients end up having difficulties with initiating movements, slow movements, and more of that Parkinson's phenotype. And in Parkinson's disease, actually, the basal ganglia is also affected. Specifically in Parkinson's disease, it's the dopamine inputs from the substantia nigra that are affected, reducing the dopamine signaling, and that also leads to that akinesia that you see in Parkinson's disease as well. Okay. So in Huntington's disease, with Huntington's disease, even though we've learned a lot about the genetics of the disease and some of the underlying neuropathology, there's still currently no cure for Huntington's, and patients suffer from debilitating symptoms as well as death from this disease. So the ultimate goal of research is to understand the mechanisms of this disease better in the brain, to develop therapeutic tools that can either cure or slow the progression of disease, which would be the ideal outcome, or reduce the impact of symptoms on quality of life. One great way to look at mechanisms of disease and cellular mechanisms is to use animal models, and a variety of models have been developed for HD. This includes everything from fruit flies and worms, all the way up to large mammals like sheep and monkeys. Specifically for my work, I focused on a mouse model, and mouse models have by far been the most popular model to study Huntington's disease, with dozens of them being created over the past 50 years. And since the gene for Huntington's disease was discovered about 30 years ago, many genetic mouse models have been created. So in a genetic mouse model, part of the gene that contains the Huntington mutation from humans will be inserted into the genome of a mouse. And then from there, that mouse will be bred for many generations until you get consistently expression of the Huntington gene in the mice. And then in my research, we would breed a wild type mouse with a Huntington's mouse for all of our studies. And this gave us not always exactly 50-50, but on average, 50% of the offspring would be wild type, 50% would be Huntington's. This is great for research with our control group because our control group can then be the litter mates of the Huntington's disease group, which can get rid of other confounding factors like the environment that the mice are raised in. So for all of my research, I'll be comparing the Huntington's animals to their litter mates. So in vitro experiments are one of the main ways that Huntington's disease has been studied. And just a quick definition of in vitro versus in vivo. So in vitro experiments refer to studies that will look at things like cell cultures, so isolating cells in a dish, brain slices from animals, electrophysiology studies where you stimulate and record from neurons in a brain slice or in a cell. And this is a great way to study the cellular mechanisms of disease. In HD research, in vitro experiments have revealed changes to neural signaling that start at early stages of disease and may be contributing to neurodegeneration. Some examples include changes to neurotransmitter signaling, synaptic plasticity, mitochondria function, and more. So all of that work, or most of that work was done in vitro. And I guess I forgot to define this earlier, but in vivo would be when we're looking at the whole organism. So the mouse is still alive and we're doing experiments in a live mouse. So for this work, I was interested in whether changes that have been discovered in vitro can be observed in vivo and how they may contribute to the behavioral symptoms of HD. So lots of work, as I mentioned, has been done in vitro. But when we're isolating a cell in a dish or just a single brain slice, we don't really know what's happening in the entire network of the brain. We also don't know how this correlates with the actual behaviors of the animals. So I had two main questions for this project. How does neural activity in the dorsal striatum encode motor skill learning and spontaneous behavior? And how is this altered in Huntington's disease mice at different stages of disease? As I mentioned, there are many models of Huntington's disease, and each of them have their distinct pros and cons. The model that I used is called YAC128, and I'm just showing this slide just to express that in this top part is showing the human progression of disease, and then the bottom here is showing the mouse progression of disease up to 15 months of age. And this model recapitulates many of both the neurological symptoms of the disease as well as behavioral symptoms. Some of the behavioral symptoms include hyperactivity at early stages, hypoactivity at late stages, cognitive deficits, as well as motor learning deficits. I had male YAC128 mice at 2 ages for this study. The first age was 2 to 3 months, and this is considered early or prodromal. So similar to that prodromal stage I talked about at the start, 20 years before chorea onset. In the case of my study, I'm calling this the pre-manifest stage. And I compared these to their wild type litter mates. And then 6 to 7 months, which I call the manifest stage, where they're showing overt motor symptoms at this stage versus wild type litter mates. And then I looked at 2 behavioral tasks. The first one is the accelerating rotarod. So for this task, mice are placed on a rotating rod that gradually increases in speed, and they learn to stay on the rod as long as they can. And the standard outcome measures that are used in the literature are latency to fall, which is a measure of motor coordination, as well as the change in latency to fall over training, which is a measure of motor learning. As they do the task multiple times, they get better at staying on the rotarod. I also looked at the open field, and this is a task where the mouse is placed in a box and they are able to explore the box for a set amount of time. Some outcome measures for this task are total distance traveled as a measure of their activity levels, and center time, which is a measure of anxiety-like behavior, The idea being that a mouse that spends less time in the center is more anxious, and they're spending more times at the edges. And then while the mice perform these behavioral tasks, I did some in vivo imaging of their brain. So the way I did this is using a common imaging technique in the animal world called calcium imaging. And I specifically use a calcium indicator called GCAMP. So the premise for calcium imaging is that when a neuron is activated and it has an action potential, one of the things that happens is an influx of calcium into the cell. And GCAMP, in the case, this is the calcium indicator that I'm using, is a fluorescent molecule that has GFP, green fluorescent protein. And normally it is very dim, but when calcium enters the cell, GCAMP is expressed in that cell, then you get fluorescence of the signal. So you get an increase in fluorescence showing activity of that neuron. So this is an example of some cells that are expressing GCAMP in the hippocampus in this case. And this is an example of a signal that you'd get with this increase in fluorescence during an action potential. And I'll show you a video so you can really see what this looks like. So this isn't my work, but this is the work of another lab, and they're imaging in vivo in a mouse in their visual cortex while they show the mouse different gradings, so different images that you'll see up in this corner up here. And then the cells are expressing GCAMP. So you can see these flashes of activity every time these gradings are shown to this mouse. So that's GCAMP. And for my study, I used a different technique called fiber photometry to measure GCAMP activity in the brain. So with this technique, a fiber, an optical fiber, is inserted into the brain of the mouse. And for my study, I specifically inserted it in the striatum. So this is the striatum in the mouse. And I had GCAMP in the right striatum of the mouse. This optical fiber then recorded the calcium signals from the striatum of the mouse while they were behaving. So this is an example of the mouse. This is what they normally look like when they're just hanging out in their cage before an experiment. And so this, if you can see that here, is the connector here. For the experiment, they're connected to our photometry system through this patch cord here, and that's how we get the signals from their brain. So for these experiments, I had a specific type of GCAMP called JGCAMP7F expressed in all of the neurons in the dorsal striatum. And I had a fiber optic cannula inserted there as well to record GCAMP activity. And just a flow chart of how this experiment goes. So the GCAMP is injected into the brain and expressed in the neurons at the same time as a fiber is implanted. And then after about 3 weeks, giving time for the mice to recover, as well as time for the GCAMP to be expressed, which can take a few weeks. Then we habituate the mice so that they get used to being connected to this fiber, because this is a technique where we can measure them during freely moving behavior, but it still takes some time for them to get used to being attached to the system. After this, we do the behavior. So that was open field and accelerating rotor rod for 4 days. And then at the end, we also check the placement of the fiber in the brain just to make sure it's in the right place and that we have good GCAMP expression there. So here's an example of what this looks like in vivo. So this is the back view of a mouse performing the rotor rod. And then this is live GCAMP activity in the brain at the same time. And again, this is in the striatum. And this is a wild type, one of the young wild type mouse from the young group. So here I'm showing you the open field. So this is what the mouse looks like when it's just walking around in the open field. And then this is an example of a single open field trial, all of the GCamp activity that we collect over 10 minutes. So you can think of each of these peaks as a group of neurons firing. So larger peaks are corresponding to more neurons firing in the striatum. And then here is an example of an entire rotarod trial here. So this is the GCamp signal before, during, and after the trial. And before and after the trial, we have the mouse just freely moving in an empty cage. So they may be walking around, sniffing, doing mouse things in that empty cage. That's why you still see activity happening in the striatum. And then when they're on a rotarod, here the black line is when I pick them up, and then this black line is when I put them back down into the empty cage. And one thing you can kind of appreciate here is that the activity in the brain when the mouse is on the rotarod does look quite different to their freely moving activity. You get kind of these smaller, more frequent peaks of activity while they're running on the rotarod. And I'll talk a little bit more about that in a few slides. For the rotarod protocol, I had 4 days of training where the mice performed 3 trials per day. So they had 12 trials total. Each trial, the rotarod accelerated from 5 to 40 rotations per minute for a total of 300 seconds or 5 minutes. So if they made it to the end, then they just got to come off the rotarod. They didn't have to fall. In my pre-manifest group, so the 2 to 3 month old mice, I didn't find any difference in the latency to fall on the rotarod. So they performed similar to wild type according to this measure. But when you look at the manifest, so 6 to 7 month old mice, as I expected, these mice shown in red here are severely impaired on the rotarod compared to controls. Then next, looking at the activity in the brain. So here's another example of the rotarod. This is specifically a wild type on day 1 of training. And one simple way that we can analyze this data is by averaging the activity during the different parts of the trial. So the before, rotarod, and after, and look at how that changes over training. So you can see here that on day 1, the overall activity while the mouse is on the rotarod is a lot higher than before and after the rotarod. And then this looks a bit different by day 4. So one thing you'll notice is that the rotarod trial is much longer because the mouse has learned to stay on for longer. So the duration of the trial is longer, but also the pattern of activity changes, where they do have an initial increase in activity when they first get on the rotarod, and then this decreases to these small frequent peaks of activity while they're performing the task. And this was something we saw pretty consistently over learning, that the activity was a lot higher on day 1 and then reduced over time as they learned the task. So just focusing on wild type here, this graph here is showing latency to fall in gray. So that's increasing over the 12 trials. And as this increased, the average GCamp activity in the striatum decreased over the days of training. So there's this inverse correlation between their learning of the task and the activity in the striatum. And coming back now to the YAC128. So as I mentioned before, the pre-manifest YAC128 didn't show a change in their behavior according to latency to fall. But interestingly, they had reduced activity in the striatum specifically on day 1 of training. So even though their behavior looks similar, there's something different already happening in the striatum in these mice. And just a couple examples here to highlight this point. So I have 2 different example animals, and I've averaged their activity on the first day of training during the rotarod task. So the gray is when they're on the rotarod. And you can see in the gray trace here that the wild type mouse in this case, it has higher activity than the red YAC128 mouse. Whereas in these same mice by day 4, the activity is a lot more overlapping between the two animals. So as I mentioned, the overall activity seems to have this inverse correlation. So latency to fall increases and the average Z score on the rotarod, which is our measure of neural activity, decreases. If you look at a correlation between these two measures, their behavior and their neural activity, there's a strong inverse correlation between the two. This is in wild type. When you look at this in the YAC128 mice, we found something interesting here. So one thing you'll notice in red here, showing the Z score on the rotarod, is that this was decreased in the YAC128 mice, just like I showed in the last slide. So their activity in the striatum is decreased early on in training. And then when you look at the correlation between the two measures, latency to fall and striatal activity, there is still a negative correlation. But when you compare these two correlations, they were significantly different. So the early stage YAC128 had a significantly weaker correlation between the behavioral results and the neural activity. So there is some kind of disconnect here between what's happening in the striatum and the behavior. So this brought me to be interested in other measures of motor performance on the rotarod. So I was interested in how they're performing the task beyond just the latency to fall. To do this, I used a machine learning program called Deep Lab Cut to track the position of the mouse's paws compared to the top and the bottom of the rotarod. And so you can see that here, Deep Lab Cut is labeling the right and left paw as well as the top and the bottom of the rotarod at 0 and 3 centimeters here. And here's an example of what this looks like. So on the y-axis is the Y position, the vertical position of the paw. And then this is time over an entire rotarod trial. So as the rotarod increases in speed over the trial, you can see that their step size gets larger and larger. And they also have more of these slips where their paws go below the bottom of the rotarod. And one thing I noticed between the different genotypes is, so here I'm showing, I noticed some differences in the way they're performing the task. So this is a wild type, and this is a Huntington's disease mouse. And both of these mice stayed on the rotarod for 300 seconds, the entire duration of the trial. But you can see the Huntington's disease mouse is having a lot more trouble with the task, and they're slipping a lot more, and overall look less coordinated compared to the wild type mouse. And then this is captured here with the Deep Lab Cut tracking. So again, this is time, and this is the position of the paws. The wild type looks pretty good. The steps are getting larger and larger as the rotarod accelerates, but they still look a lot better than this YAC128 example, which has a lot of these big slips below the bottom of the rotarod and overall more variability in their step size. So I explored this in the YAC128 mice, and this is all actually with the younger mice that didn't show any changes in latency to fall. And what I found is if you look at the paw height over a single trial on day 1, the both of the mice kind of slowly reduce their paw height over time, and especially at the end, the YAC128 have even lower paw height than the wild type mice. Whereas by day 3, looking at paw height over time, it's a lot more condensed, a lot less variable. And the genotypes, there's not much of a genotype difference, maybe a slight difference in the YAC128. If I look at just the mean paw height over time, over the 4 days of training, their overall paw height increases as they get better at staying on the rotarod. And then looking at the standard deviation in paw height, so a measure of variability in their step size, this reduces over training. And interestingly, this was significantly higher in the Huntington's mice. So they had a higher variability in their paw, in the step size compared to the wild type. And then the last thing that I looked at with the rotarod task is something that I called foot slips. So what I did is every time, using the deep lab cut tracking, every time the mouse's paw dipped below the rotor rod, I counted that as a paw slip, where they're kind of almost falling off, but then saving themselves and staying on the rotor rod. And I looked at the difference between this in wild type mice compared to YAC128 mice. And overall, in this example here, you can see that this YAC128 mouse is having more of those paw slip events than the wild type mouse. And I've pointed to a few here, but there's even more than I'm pointing to here with the arrows. If we quantify this, we find that early stage YAC128 have a larger number of paw slips per minute compared to wild type mice, specifically on day 3. And I've isolated day 3 here to show the big difference between the genotypes on day 3. And this graph here is just showing over a 300-second trial, the cumulative number of paw slips over time. And about halfway through the trial, the number of paw slips kind of skyrocket for the YAC128 mice, but not for the wild type mice here. So this was a really cool result, because if you just look at latency to fall, it looks like the Huntington's mice are performing the same as wild type. But when we start looking more closely at their paw kinematics, we can see quite a bit of differences coming out in their behavior. And then the last thing I did is I looked at the pattern of neural activity in the striatum surrounding the paw slips and how this changes over learning. So I specifically isolated the left paw slips for this, because I have the fiber measuring activity of the right striatum, and activity in the right striatum is usually correlated with activity in the left side of the body. And I found some interesting results with this. So if I isolate just the first 2 days of training, the overall slant or ramp of the activity is going down, and that's partly just the nature of the activity on the striatum. The overall activity in the striatum reduces while they're on the rotor rod. But when you look at the difference between the wild type and YAC128 here, you can see that nothing really happens specific to the paw slip. And sorry, I'll just explain that this line here is when the mouse's foot dips below the bottom of the rotor rod. So I've lined up all of the activity from all of the trials to every time the mouse has a paw slip. So in wild type, not that much happens specific to the paw slip on early days of training. But in the YAC128, we see this elevation of activity right after the paw slip happens. And then it gets really interesting when we look at 3 and 4. So later on in training, when they've learned the task, the wild type mice show this increase in activity right before their paw slips below the bottom of the rotor rod, that then reduces after that. And this is a very consistent result surrounding the paw slips in wild type animals. Whereas in YAC128, instead, we definitely don't see that increase in activity, and instead maybe even a slight dip in activity right after the paw slip happens. So this is just summarized with this graph at the bottom here showing activity on day 1 and 2 versus 3 and 4. But this was a really interesting result, potentially some kind of neuroplasticity, a change in signaling in the brain in the wild type mice that is perhaps helping them learn to respond to the paw slips that we're not having in the YAC128 mice. And then just quickly, I also analyzed the rotor rod performance in the older mice. So these are the manifest HD mice. And these mice, as I showed before, had a severe deficit on the rotor rod when we looked at latency to fall. They also have severe deficits in other measures. They have an increased frequency of paw slips below the bottom of the rotor rod and changes to the paw height. So they have a reduced paw height compared to wild type and an increase in the variability in their paw height. This says left paw, but this was also shown in the right paw. So these were so bad at the rotor rod that we actually separated them into 2 groups. So I noticed that about half, actually exactly half, of the YAC128 mice were so bad at the rotor rod they didn't learn it at all, and they would fall off within a couple of seconds. No matter how hard I tried, I couldn't get them to learn the task. So I ended up separating them into poor performers and good performers. Even the good performers still show a deficit in their latency to fall compared to wild type. But I separated them this way because if they're falling off within a couple seconds, I can't really look at neural activity associated with the task when they're not performing the task. So when I looked at the striatal activity, I just looked at the good performers. And here I found an interesting result. I'm kind of opposite to the young group. I found this increase in activity in the striatum of the Huntington's disease mice compared to the wild type mice. So these mice are severely impaired on the task, and they have much more activity in the striatum while they're performing the task. Here's an example of the activity during a trial on day 1 with the yak in red compared to wild type, compared to day 4. And by day 4, they look more similar again, but they just have this high activity on day 1 of training. They're also much more uncoordinated on the rotarod. And so I think that some of that uncoordination during the task could be causing this increased activity in the striatum. Okay, so last thing I'll talk about, so switching gears, just very quickly talk about their activity in the open field. So the other task that I had them do was exploring an open field. They did 10 minutes a day exploring the open field for 4 days. And I didn't find any differences in kind of the standard behavioral measures in the young mice. Their total distance traveled and center time and other things were similar. In the older mice, though, I found an interesting difference. Their total distance traveled, so this is a measure of their activity level in the open field, was similar in the older HD mice compared to wild type. But when we look at the change in the distance traveled, so we compare day 1 to the other 2 days, wild type show a decrease in their distance traveled because it's a familiar environment. So mice will get habituated to the environment and they won't explore it as much. Whereas the YAC128 mice don't show this change. So this is seems to be some kind of memory deficit in these mice where they're still exploring the open field just as much as the first day. As far as neural activity goes, I analyzed this data a little bit differently than the rotarod data. So I looked at peak frequency and other measures of these peaks of calcium activity in this data. And what I found is in wild type mice, during a task, the peak frequency, so this would be the number of peaks that happen per minute during the task, correlated positively with the total distance traveled. Since the striatum is important for movement, it makes sense that more neural activity would correlate with more traveling around the open field. This was in both the young and the older wild type mice. But then interestingly, in the YAC128 mice, I didn't find this correlation. So even if it didn't matter how much they were traveling, it didn't correlate with the neural activity in the striatum when we look at it this way. So there's, again, some kind of disconnect between the activity in the striatum and the behavior that we're seeing. And this is both the young and the old mice. So to look a little bit more closely at their behavior in the open field, I looked at specific categories of behavior. I just looked at 3 broad categories. So one of them is being active. So this is when they're locomoting around, so walking around the open field, and rearing, which is when a mouse goes on its hind legs, kind of stands up on its hind legs. Rest, so when they're not moving, and then grooming behavior. Just an example here. You can appreciate that the activity is a lot higher in this example when a mouse is active versus when it's resting, where you might see a few small peaks, but not this extended activity here. And then also we would sometimes see these peaks of activity right when the mouse starts grooming. And then one thing that I found when I looked at the number of peaks that happened during these behaviors is that there was increased peak frequency happening when the mice were at rest in the Huntington's disease mice. So both at early and late stages, so pre-manifest and manifest mice, there's this trend for increased peak frequency when they're not moving at all, which might be also the reason that the correlation is affected between their total distance traveled and their neural activity, just this baseline increase in activity in the Huntington's mice. Okay, so in summary, YAC128 mice show progressive paw kinematic deficits on the accelerating rotarod and changes to striatal activity during the task. I found in both age groups that there was increased paw slip frequency and variability in paw height on the rotarod, and these impairments worsened with the disease stage. I found a weaker correlation between striatal activity and rotarod learning in the pre-manifest YAC128 mice, changes to striatal activity associated with paw slips in the pre-manifest mice, and elevated activity during early stages of learning in manifest mice. And then I also found that YAC128 mice showed disrupted striatal activity and behavior in the open field. So both pre-manifest and manifest YAC128 mice did not show a correlation between total distance traveled and the calcium peak frequency in the open field. And pre-manifest YAC128 showed a significantly higher calcium peak frequency at rest. So future directions of this research, one thing that my lab is currently working on, my previous lab, from some experiments I did before I left, was looking at specifically the D1 and the D2 pathway that I talked about, so the go and the no-go pathway in the striatum, to understand the role of these two pathways in motor learning and behavior, and also how these two pathways contribute to the behavioral symptoms in the Huntington's disease mice, as well as looking at the open field behavior in more detail using some other machine learning software. So we can use some of the software to examine the different behaviors in more detail, as well as the kinematics of the paws during these behaviors. Here is just an example of some preliminary data looking at both pathways at once. So in these experiments, we're using fiber photometry again. We have a green version of GCamp in the D1 neuron, so the go pathway, and then a red version in the D2 neuron. So we're able to image both of these neuron types at the same time during behavior. And this is a rotarod task. So between the black lines here is when the mouse was on the rotarod. This is a single mouse. And one thing you'll notice is that both of these pathways are often active at the same time. So as I mentioned earlier, more recent research has shown that it's not simply a go and a no-go, but it's these two pathways kind of work together to refine movement. So both pathways seem to be active during the rotarod task, but we're interested in how the specific patterns of activity differ during the rotarod and different behaviors. Okay, so that's everything. And just some acknowledgements. I wanted to thank my PhD lab, my supervisor, Dr. Lynn Raymond, and everyone in the lab that helped out. The Murphy lab at UBC also helped out, as well as the DMCBH, the Center for Brain Health at UBC, helped out a lot. And then I also, when JD Baines used to be at Calgary, I came here years ago to learn about fiber photometry. So they helped us with setting up our system. And then thank you to all the funding sources, and thank you for listening.
Awesome. Thanks so much, Ellen. Do you have any questions to get us started?
Well, first of all, that was great. Great work. Thanks for presenting. How do you get the optical fiber exactly where you want it? Is it like an image guided thing, or do you guess?
Yeah, we use the brain atlas to find, so we have like a mouse brain atlas that shows us and we use that to get the coordinates. When we first started, there was a lot of trial and error and practice to get the right location. Luckily, the striatum is very large in a mouse brain, so it's hard to miss it. But yeah, so it's basically trial and error the way that we do it, using the mouse atlas as a guide. It tells us the coordinates, like the Y and X coordinates. And
is there like some way like you're like, oh, it's in there, or do you have to like use an image or something to ensure that it's where you want it?
Oh, so afterwards we check all of them after the fact. So afterwards we'll take brain slices from the mice, and then we'll take pictures of it to see where the fiber was, because we can see the fiber is large enough that we can see where it was after we take it out of the brain.
Okay.
Yeah.
Got it. And then another quick one is, what's the difference between these bad performers and good performers?
Yeah, that's a good question. I'm not sure why some of them perform so badly compared to the others. I looked at some things. One thing that happens in the YAC128 model is they gain weight, which is it's one of the problems with the model. So I thought maybe, because if you're heavier, you can be worse at the rotarod task because it's harder to stay on. But there's no correlation there. So some of the bad performers were heavier, some of them weren't. And so that wasn't what was happening. I'm not really sure why, because they're genetically the same.
And that means that the severity of the disease would be the same?
It should be, but we still see variability in mice, even when they're genetically the same, we still see variability. So one thing is, in our experiments, the mice, particularly the older mice, perform worse in general on the rotarod. Even though it's fiber photometry, it allows freely moving behavior, it's still a little bit harder for them, having the fiber kind of sticking out of their head, to perform the task. And it seemed like the older group performed even worse than we expected on the task. So in those poorly performing animals, maybe for whatever reason, the fiber is affecting them even more and making their performance even worse. But yeah, it's a bit of a mystery. Yeah.
Just to add a question about the fiber being embedded, do you find that there's much scarring or any issues with that? Like if you have this thing like jammed into the brain tissue, like do you have any issues with like tissue, like in terms of measuring the fluorescence and like healthy cells and things like that?
Yeah, I mean, there's definitely damage to the brain just from inserting the fiber. It is pretty small, so that minimizes the damage. But when I look at the brain afterwards, because we take images, you can see sort of darker areas immediately around the fiber and then brighter areas further away. So some of the cells, I think, immediately around the fiber are definitely damaged. But we still get a large enough signal in general.
Would that healing or like those kind of processes differ between the wild type and the
we don't think it should differ. We didn't notice any difference when we looked at the slices afterwards. It is possible though. It's something to keep in mind, but we don't really have any reason to think that it would be different between the two.
Do you notice the difference between the bad performers and good performers in the box, like compared to how they move around the box?
Yeah, I did look at that, and I didn't see a difference there in their behavior. So yeah, because I thought maybe they'd also be hypoactive in the box as well, but I didn't see a difference there. Yeah, nothing significant between the two.
Yeah, question in the back.
Is it possible that the good performers and poor performers differ in the number of CAG repeats that they have?
In this model, they shouldn't. So this model is called YAC128 because it has 128 repeats.
Okay.
So rodent models have many more repeats than the human condition, just because it's harder you just need more repeats to get the behavioral symptoms in the rodent model. That is a really good question though, because we use another model called Q175, and that one is susceptible to spontaneous expansions of the repeat. So that model actually started out with 140 repeats, and then it expanded to 175. And now we found in our lab that usually they had like 183 repeats. So we did start to control for that and make sure we had a certain number of repeats. But this model isn't susceptible to that, so it should be stable at 128. Yeah.
So has anyone looked at like aging up the young ones, so to see how they performed when they were doing the task young, and then how they also performed later to trajectory to see?
Yep. Yeah, that's a good question. Lots of people, other studies have looked at that. There is a learning effect where if they did learn it when they were younger, they'd perform better. So if they learn it at 2 to 3 months, then they will remember at 4 to 5 months and so on. Then an animal who's never done the task and then learns it for the first time. But yeah, they have looked at that and found deficits that get worse. Even though they've learned it before, they still found the deficits worsen with time in the older animals.
Any other questions? Yeah.
Thanks, Ellen. I was just curious about your fluorescence recordings, where you had it before, during, and after the drug test.
Yep.
Just had a couple of questions.
Go back to that.
Yeah, sorry.
Like
Yeah,
here,
that-
right?
So I'm just wondering, like, you have them, you're picking them up and putting them on the road during that
Yep.
window. Is that
Yeah.
suppression that occurs before you put them on there? Is that a real thing? Like, does it kind of like see your hand coming and shut off or something?
Yeah, like right before the
Yeah.
pickup. I just realized I'm supposed to repeat the questions, right?
Oh,
So I just realized I haven't been doing that. But yeah, so the question's about the suppression of activity right before you
it
pick them
seems to
up.
be in a few different traits.
Yeah, so I mean, one thing it could be is that they're getting scared and they're stopping like
Right.
right before I pick them up. So, you know, the striatum is so much of the activity is movement related. So if they're less active, then there's going to be less activity. So my
Well,
best
day
guess,
4, it's like they don't care so much.
by then they're more used to
Yeah.
the whole process probably because day one, like the first time they do it, it's their first time going on the rotor rod while it's moving. Yeah, so that would be my best guess of
Yeah.
why it suppresses right before. Yeah.
And then you looked at frequency for the open field test. And I was
Yeah.
wondering if you looked at any frequency stuff here.
Yeah.
Peak or mean frequency. 'Cause
I
it looks like there's clearly something going on.
Yeah. So the question is, did you look at frequency for
Yeah.
the rotor rod? I looked a little bit at frequency. I didn't find any, like I thought I would find something
Right.
cool between the genotypes, but I didn't. So it didn't end up getting included in the final manuscript. But one thing that definitely happens on the rotor rod is the peaks get, I also looked at peak amplitude and width, and they get smaller amplitude and smaller width. So they get faster, these smaller fast peaks while they're on the rotor rod compared to before and after. Yeah.
Online question, Noah?
Okay, we got an online question here, so
Yep.
I'm gonna read it off. While it is agreed that the rotor rod test of the open field tests primarily reflect gross motor behavior and to some extent the phenotype of HD, they do not seem to be specific to unique movement deficit like chorea. Observing this moment, a movement of the palsy to get a bit closer to phenotype. So given this, I'm curious if it would be possible to conduct behavioral assessment that focused more on fine motor skill, especially when observing the accompaniment body.
Right. Yeah, that's a good question. And another kind of problem with the rodent models is they don't show chorea symptoms. So it's just not something that we can get in the mice, at least not when we use these genetic mouse models. But the question specifically asking about fine motor tasks, and there are other fine motor tasks that have been used in Huntington's models. My previous lab, the Raymond lab, has done some tasks where mice learn to pull a lever, so we can look at fine motor skills of the upper body. And this is similar to some of the deficits that happen in people with Huntington's disease, with deficits in their upper body movements, motor learning. So this was a motor learning task where they learned to specifically move a lever into a very specific location and hold it there and then let go. So there is newer research looking at those kind of tasks in Huntington's disease mice. Yeah.
While we're on the topic of the tests, I'm wondering how specific these findings are to Huntington's disease, and then or would you expect the same thing with other neurological diseases? Like if you had an ALS mouse or a Parkinson's mouse or something like that, would you see the same kind of thing?
So the question is, how specific are these tasks for Huntington's?
Yeah.
Other mouse models show these deficits. So rotor rod is widely used with lots of different mouse models. This is a very good task. Huntington's disease mouse models are very, all the different types seem to show these rotor rod deficits, so it's good for Huntington's disease. I am pretty sure that Parkinson's disease mice also show deficits on the rotor rod. I would expect them to, because also Parkinson's disease patients show motor skill learning deficits as well. I know other models, I'm not sure about ALS mice, but I know other models of different motor symptoms. I know stroke models also show deficits. So I would expect definitely not specific to Huntington's disease. Yeah.
Just a question in terms of on your last slide, you had the go versus no-go pathways, and you said it's like more complex where you have like activation at similar time points for both of those pathways.
Yeah.
But if like, presumably the time that you're looking is quite large, but if you were to zoom in on like a single step, would you see that there would be like a correlation between the activation of these different pathways and like limb acceleration or things like that? Like there's some, like if you were to look at a finer time course that they wouldn't be on at the same time?
Yeah, so that is a great question. That's what I'm hoping we can do. And my previous lab is kind of working on analyzing this right now. But other labs, when they look at like exactly like what you're saying, very specifically at like right on the onset of a step, or even just the onset of a behavior like starting walking, then they'll see different patterns of activity in these two pathways. There's one paper, Sabatini is one of the final authors on it, but they looked at activity in both of these neural populations during many different behaviors and found different patterns, like the start of a movement, the D1 would be active, but the D2 wouldn't be. Turning, the D2 seemed to be more active during turning behaviors, like the initiation of turning. So one thing I really want to look at with this is during a foot slip, what's happening in the activity.
So would you expect for like the Huntington's model mice that you would have that lack of pre-activation that you saw in the slips from the GCamp or the fluorescence? Would you expect in terms of your pathway activations that you would have differences between the wild type and the Huntington?
Yeah, I would expect that. I'm not exactly sure what they would look like, but I think, yeah, something like with the foot slips maybe where there's less activity during the initiation of a movement maybe. The older Huntington's mice showed that memory deficit in the open field. So maybe there would be something different over time in the open field where the activity is increasing in one pathway and not in the other pathway. I'm not exactly sure what to expect, but I think we'll find some interesting results with that.
Yeah, cool.
Yeah, I got a question now. Like, go ahead, Walter.
Hey, yeah. Thanks, Ellen. Can you hear me?
Just 1 second. I was turning up the volume.
Okay, can you hear me, Ellen?
Yep.
I think so.
Oh, good. Yeah.
Oh, yeah, there.
Good. Yeah. I didn't hear all the questions, so hopefully I'm not repeating some of the questions that were already asked. Well, thanks for a very interesting talk. I was just interested in the disease first. Can you tell me what the prevalence is of Huntington's disease?
Sorry, I missed that.
The prevalence.
Oh, prevalence.
Prevalence.
Yeah, so it's about 5 in 100,000 people worldwide, and then it's more common in Western populations. So I think it's about 13 in 100,000. It's the most common inherited neurodegenerative disorder. Yeah.
And then you said that these people die about 15 years into symptoms of the disease. So why do they die?
That's a good question. So the actual cause of death, there's different causes of death, but the most common is actually it's aspiration pneumonia. So at very late stages of the disease, they start to have problems with swallowing, and then that can sometimes lead to, I guess, some food getting into the lungs, and then they actually die from pneumonia. Other causes of death could be from falls. So complications after a fall, and actually suicide is more common in people with Huntington's than the general population as well. So there's a variety of things that cause the death or that are kind of complications from the disease.
And more a technical question, you have the slide up there, you know, with the green fluorescent and the red fluorescent. So how do you get this into the specific cells? How do you know that the green goes into the D1 and the red goes into the D2? How do you do that?
So for these studies, we inject They're genetically expressed into the different cell populations. So just briefly, there's a bacterial plasmid that will have the GCAMP, and then it will be driven by a promoter. So in our case, we're either using the D1 promoter or a D2 promoter that specifically expresses them in the cells. And then we're also using some staining afterwards to make sure that they're being expressed in the correct cells as well.
Great, that's really neat, yeah. And the other thing, has anybody ever looked at what happens to the muscles in these people, like the structure of the muscle? Does it change in any way?
Yeah, I'm not an expert on the muscles, but I do know a little bit about that. I know people with Huntington's disease lose weight, so they definitely have muscle wasting. And so they lose muscle mass over time. And I don't know much more about like the mechanisms behind that. I know that mitochondria are involved. So there's it actually both in the neurons, there's mitochondrial dysfunction as well as in the muscles in Huntington's disease.
And have you or anybody else ever tried to measure muscular activity when these people are, when these animals are on the rotating wheel, and you measure brain activity, and I'm kind of interested how that might translate into muscle activity? Has anybody ever done that?
That is a really interesting question. I'm actually not sure if anybody has done that. The rotor task it's very common, so I feel like somebody might have done that before. I'm not sure, but that would be something interesting to look at.
And then just last, a very technical question again. When we make calcium signal measurements, for example, in cartilage, the calcium signal stays on for a very long time, like the flash comes and then stays on for 10, 15 seconds. And so is that different in the brain? Do you get very short flashes, or does it also stay on for a certain amount of time? And so you get an accumulation of signals?
Yeah, so that is similar in the brain. So it's one of the downfalls of using a calcium imaging is the time course is a lot slower than an action potential. The initial rise time is pretty good in calcium imaging. It's pretty quick. I think like 20 milliseconds, but then the decay is just longer. And that's to do with the kinetics of GCAMP and calcium. The more recent GCAMPs they're continually trying to improve upon that, but it is a problem in the brain as well.
Good, fabulous, thank you.
Yeah, thank you.
Do you have any more questions? No? All right, so I'll thank our speaker again.